see y'all eating this stuff too. I talk about it all the time. I'm like, I told him the other day again, I'm like, I'm so proud of y'all. Because you don't follow my eating style. It takes so much work to make it healthy though. You know, I watch them, I'm like, what are you making? You know, it's just odd. My daughter walked in yesterday with a can. It said garbanzo beans. And I said, she said, are these chickpeas? And I said, well, they look like chickpeas to me. I mean, if they were in something, I wouldn't eat them because I don't like chickpeas. I'm pretty sure those are chickpeas. Those are chickpeas, right? You know, and then she walked out there and she yelled, Google it. I don't know. I don't know who bought those. I wouldn't buy them. Anyway. All right, I only have one story for the weekend. I was on the lake LBJ all weekend, so I'm tired. I'm not tired because it was on the lake, but they had a ramp just for the boat people. And it was like a homemade ramp. There's a lot of them on LBJ. And they had poured like some concrete. I couldn't tell how deep it went there. I put the boat in there anyway. But it only had like, I had from like here to the wall, not even that far. It was a peninsula, so it was just water over there. So I had to back it in at like 90 degrees to get it in there. So I brought two people with me, stood them on the side, and said, listen, if I start to run over you, yell, but don't move. Because, you know, you ever have somebody that you put them somewhere where they're supposed to guide you and they don't say anything, and they back up when you're going off the edge? Don't back up. Yeah. Anyway, I got the boat in there just fine, so I went over there by myself and was going to pull it out. It all went well until when you're by yourself, the easiest way to do it is leave your runners out of the water a little more and drive up on there, right? And when I went up on it, one of the runners like shot out to the side, so they were rotted and broke. And so I thought, well, I'm going to have to back it out just a little further and float the back of the boat so that you can push that runner and maybe pull it out, maybe I can get it out of here. When I did that, I heard clunk. That was the transom of the back of the trailer going off the ledge. And so then I did what everybody did. I went and got help. No, I didn't. I thought, well, I'm going to have to throw it in a four-wheel drive now and just power it out of there. It went nowhere. It went nowhere. Anyway, but I happened to have a college football running back and a high school front lineman that's almost 280 with me, daughter's boyfriends, and I had them come out there and lift it up while I drove out. One of them was underwater while we did it, so I'm not going to tell you again. But it was so deep that they were up to their neck, and one of them could get it and lift it. The other one was a little shorter, and he literally, what we had to do, this is the only reason I'm telling the story, because that's not even interesting. What's interesting is to watch him, is we had it timed. I said, okay, we tried it once, it didn't work. And the guy that was short said, listen, I don't think I was lifting my size yet, because I got to drop down in the water. So he said, I'm going to go down underwater and get under and pull up. So give me 4 seconds or so, and then you lift your side, and then he yells to me. So I'm in the truck, and I say, you know, whatever, I'm in gear, and I'm ready. The other dude drops underwater, lifts it up. The other dude counts 4 seconds. And about 4 seconds, he yells go, and I didn't power. I mean, I drove forward, and we got it over. Now I got to go back out to the lake. I'm like, you got to put the boat in the water, fix the wood. Well, you don't have to, I guess. These are good problems to have, right? Right? While you were studying, these are good problems to have. I had more problems that kept me up all night one night with one of my kids, but that's a whole different story. I looked at my sleep app to see if there's a whole day missing. Like 36 hours and nothing. I figured it would at least think, because I did like doze off for like 15 minutes. It doesn't even count that. I didn't even notice. Anyway, I got plenty of sleep last night, so I'm good. Y'all don't care about whether I got sleep last night. That's funny. Yeah, who cares about your sleep? All right, we left off here. If not, correct me. I believe we did. We had just finished talking about rigor mortis, and then we broke off. Right? And so we were discussing muscle structure and sliding filament theory and how those little sarcomeres function. And I think I got the idea last week that we were pretty much tracking. I think everybody was kind of getting it. Nobody said they weren't. And we're start to finish this out. We're going to start to look at basically how we're going to make muscles contract, what are motor units again, what's motor unit recruitment, and then we roll into neuro today. It's probably just the first couple of slides of neuro. We'll get most of those neurons tomorrow. So muscular hypertrophy is what muscles, specifically skeletal muscles, all muscles do this, but skeletal muscles go through hypertrophy. So there's hyperplasia, I have a slide on it, but there's hyperplasia and there's hypertrophy. Hypertrophy just means that you're going to take everything that's inside of that muscle cell and you're going to increase them. So if skeletal muscle doesn't go through mitosis, but it does enlarge. So you're just going to create, rather than, once again, like I always say, rather than make a list, think, okay, well, what's in a muscle cell? Sarcomeres, mitochondria, everything that's in there, we're going to increase the amount of, and that will increase the size of that individual muscle cell, and over time, the whole muscle itself. You pump an iron, and you'll build up that muscle, and it'll get larger. But actually, what you're doing is enlarging every little muscle cell by putting an increased demand on it. It will respond. Right? Or it will do the opposite if you don't put that demand on it. Right? If you take the elevator every day, the legs will suffer. Right? So that's that whole slide and just kind of my talk. The actual words you use, like achieved by forceful, repetitive muscular activity, very capable of more forceful contractions. But we're also going to see, or because they contain more myofibrils, in other words, more sarcomeres, but we're also going to see that you achieve more efficient motor recruitment when you start working out, so that you can use the right motor units. And there's your definition of hyperplasia. Hyperplasia means you're going to increase the number of cells. So if cells go through hyperplasia, they are going through mitosis. They're creating more cells versus just enlarging the cells. And atrophy is when I said you do the opposite. You're going to start to, if you don't put the demand on the muscle, it doesn't need them, it will start removing, not removing components, but decreasing the amount of mitochondria or decreasing the amount of myofibrils because it doesn't need them. So the opposite of working out. And that's where I came up with, and I've mentioned several times, that in skeletal muscle, when you start to really enlarge, it's and actually somebody mentioned that when I first said it, what causes skeletal muscle growth and when, and they said, well, at puberty, and that's true. What I was saying at the time was you're born with all the skeletal muscle you have. At puberty, the hormonal changes start to cause an increase in the size of those muscle cells on its own, and then if you start to work out, put demand on the muscle, it will increase the size. All right, so a lot on that, but I just find that interesting. In muscle cells, we have myoglobin. Hemoglobin is found in the blood. Myoglobin is very similar. It's just another protein specific to muscle cells. Right? It's just like hemoglobin does. Myoglobin binds oxygen within the muscle cell itself. Right? It's one of the reasons we shouldn't see like myoglobin in urine. It should be confined to the muscle. So if you're going to put a large demand or an aerobic demand on muscle, you're going to need more oxygen. So it's helping to keep that oxygen there. So we're not biochem, but we always have to keep learning where does the energy come from and why do we need it. So when we use ATP in muscle contractions, one of the things, actually not just in muscle contractions, but we're talking about it here in muscle contractions, ATP is what drives active transport. So anytime you see sodium potassium pumps, we know they're going to use ATP. But what we're about to get into is great. You've taught, or we've learned, that ATP drives sodium potassium pumps. There's also other types of ways for, let's say, sodium and calcium stuff to get in and out of a cell that can use active transport processes. I'll get more into that. Calcium pumping ions. So just like a sodium potassium pump, we have calcium ion pumping. And I failed to mention this the other day because I normally talk about it here. But before we even get there, remember the muscle cell has a sarcoplasmic reticulum that's full of calcium. has calcium is 10,000 times higher inside of the sarcoplasmic reticulum. And I said, well, remember, let's just think of it as in a muscle cell, all of the calcium. That's not technically true, but let's just say all of the calcium in a muscle cell at rest is going to be in the sarcoplasmic reticulum. And then when an action potential comes along, it will cause depolarization, which will release calcium. And calcium goes to bind to troponin. Remember all this from last week? Starts that. Right? But can the calcium stay just out there in the cytoplasm of the muscle cell? No, because you'd be in rigor mortis, right? It would continually cause contractions. So you got to get it back into the sarcoplasmic reticulum, calcium ion pumps, which would use ATP. Because if it's 10,000 times higher in the sarcoplasmic reticulum, so we're going to, every time we dump a little bit out, we're going to put it back in. We're going to need energy to do that. We're going against a very large concentration gradient. So there's some slides coming up on that later, but we just discussed it. Cool. I'll mention it there again. And then when we looked at sliding filament theory, remember in that whole circle of how a muscle contraction occurs, you have myosin heads that are waiting to form cross bridges. The myosin head wants to bind with the myosin binding site on actin. And that's shaped because it's energized, right? And when calcium causes troponin to move out of the way, the myosin head will form that. And in order to get it to detach, you need to bind ATP again. So it uses, and then it will immediately hydrolyze ATP again. Right? If you like biochem, right, you're going to lose a group, gain a group, phosphate groups. And so since cells, muscle cells themselves don't store ATP in large amounts, we got to see how we're going to make it. And another way, better way to say that is, you're not immediately, if you start, you go to run down the stairs right now, you're not immediately going to start using our aerobic respiration. You may not need it yet. Right? In muscles, they do have some creatine phosphate that is there, which should always be inside the muscle cell. Then you can go through glycolysis or anaerobic, right? Or aerobic respiration. And you will go through those in order if you go to start running. I guess an analogy would be like you're going to use creatine phosphate for a short sprint. You might use glycolysis to go once around the track. And get further than that, or if you start taking off to run a marathon, you're going to have to continually go through aerobic respiration because you'll very quickly burn up the first two. Won't go easy. Biochem used to cover that. I'm sure they do. They give you they made you learn the time frames. Like creatine phosphate will give you 15, 20 seconds or something like that. I took the times out because I don't know how important that is unless you're studying that in exercise physiology or something. Okay, so this is where we kind of change and say, okay, we know how a muscle contracts or what it does. How do we make it do that? How are we? This is the part we kept leaving out last week when I would just say, well, an action potential causes that. So now we're going to learn, well, how do we stimulate that muscle? And how do we, that's in other words, control the contraction. So a motor unit, we're going to define it again. A motor unit is one somatic motor neuron and all of the muscle cells that it is in contact with. That's the definition, even though it doesn't really contact it. No, it's neuromuscular junction. They're spaced close. Right, when you look at one nerve impulse, it means in that motor neuron, one nerve impulse comes down that. Right? And it will cause an action potential in all the muscle fibers of that motor unit. Even when I read things sometimes, it sounds funny, but that makes sense. If we have one somatic motor neuron, and I got 5 muscle cells out here, this is one motor unit, or many motor units, that's one motor unit, and I send one action potential, or electrical potential, right, down that neuron, and it reaches the end, it will cause, right, a contraction in each one of these muscle cells that are part of that unit. Make sense? And it's always the same magnitude, right? When you look at how large of action potential travels down that. We're going to discuss that. That may not make sense right now, but if I have a motor unit and I send an action potential down it, it's going to cause a contraction in those muscle cells. But what we're going to see is that's not the max contraction. I just thought another way to look at that is that's not, let's look at one cell. Well, let's go back to our sarcomere chain. It'd be just, that's the easier way. Because if I'm a motor unit and y'all are all the muscle cells that I'm in contact with, because it's not always 5, it can be 1, it can be 20. Right? Well, let's say I'm one motor unit, but I'm in contact with the whole second row, and I send one action potential down that axon, and it reaches, it's going to cause every one of the muscle cells associated with it, the sarcomeres inside, to slide together, to contract. But it doesn't maximally do it. It doesn't contract that sarcomere all the way in. But if I send one impulse, and so in that situation, right, y'all would get a little closer. But what if I send an impulse, and another impulse, another impulse? Eventually, I could reach max contraction of the cells in that motor unit. Make sense? It's, and the brain controls that. We're going to figure that out. And what I'm saying when I said an action potential goes down there quicker or more than once, that's what it's saying here. This force of a single fiber, single muscle fiber, which is a muscle cell. Remember, that's the myofiber, muscle cell, same thing. A single fiber can produce is dependent upon the rate it's stimulated. In other words, I can stimulate it more or less. In other words, what action potentials are arriving at the neuromuscular junction? That's the connection between the somatic motor neuron and the cells. We can look at that, number, impulses per time. We're not going to calculate that out. So I give you some definitions at the end of this to go along with this, but if you have like a brief contraction of all the muscles in one motor unit, that's a twitch, or a twitch contraction. And we can measure this. You will probably never measure this in your life, unless you go work like rehab medicine, spinal medicine. I used to see myographs in the charts. Average military person just doing clinic, you're never going to use myographs. Okay? They're specific for looking at or taking and measuring force generated by the contracting muscle. And sometimes we want to know that, and like in rehab, we want to know that and see, can we increase that, and we can measure it again. It's not real important on the everyday. I get it for y'all, but for me it was because I'll give you an example. If I have somebody with a spinal cord injury, generally spinal cord injuries are not complete. We often think of, somebody damaged their spinal cord and now they can't use their legs, or they can't use their legs and their arms. Most spinal cord injuries are incomplete. Other than like ATV, motorcycle accidents, a lot of complete injuries are through the thoracic spinal cord, because they'll hit some handlebars and transect it, really traumatic. But like most people you hear, like, they say, oh, they broke their neck and now they're paralyzed. And a lot of them are not completely severed. What happens is the spinal cord is in a narrow canal, and you disrupt the bones around that area, and the swelling is going to get them. And because of that, because they're not incomplete, some of the fibers that are in that spinal cord remain intact. And so that's why we have to test, because so in other words, there are classifications. I'm getting a little off, but I think it helps to understand. Is we have these Asia classifications of spinal cords, and by the classification they tell me, I would know basically what their level is, whether they can use arms, whether they have full use, not as much use, and then I can test that. But if I take two different patients with the same Asia classification there, whatever that might be, and I look at their arms, let's say, one of them may have no use, one of them may have a few muscles they can use weakly. Well, that's a good thing. If you ever have a spinal cord, you'll definitely be focused on that. You see the movies, right? I can move my finger or something like that. The whole point I'm making here, I did a long spiel to say, I can move my finger. Okay, well, as a rehab person, can I make that functional? Can I get that muscle to come back? and yes, I can, and I might want to measure that to see the progress, versus going, I can do this versus this. You know, makes sense. I'm trying to give you all some good examples to put in your mind. Because the myograph tells you that. You take this transducer, so you take something that converts one form of energy into another. The energy your muscles are doing, and we measure that, and it comes out on a graph looking like this. This is a myogram. If you ever see one, somebody's had a recorded muscle contraction. And so you can elicit this reaction by either causing the motor neuron or stimulating its motor fibers. Most of y'all actually understand this, whether you think so or not, because you've seen a E-stem unit or a TENS, right? Where do you connect E-stem and TENS to? You're actually hitting directly on the muscle cells themselves, right? You take a TENS unit and you put it on the muscle of your forearm, right? Have you ever done that? It's kind of fun to play like you tighten them up. You're not supposed to use it like that, but if you're in the military, I'm going to do that to you once. Doesn't hurt. Just funny. But you're putting that directly on the muscle fibers. You're stimulating directly through the skin to those muscle fibers. But could I also, like an nurse anesthetist, anesthesiologist, spinal guys, could I go out and find the actual place that I can make access to the neuron that stimulates that muscle and stimulate it up here? That's what it's saying. So I can do it one or two. I can either stimulate the neuron or I can stimulate at the muscles. Of course, if you know what you're doing, stimulating the neuron is going to be a lot more specific for that muscle group. Because if I put a TENS down here, I'm going to probably get both sides of my forearm muscles, flexors and extensors. All right. So durations that we look at, I know you talked about this in cardiology already, like the refractory period. And generally in cardiology, they'll call it absolute refractory period. We're going to have to compare this graph to the movement of calcium. So if we look at latent period, this is before we've stimulated that actual muscle. And then you'll have a contraction period, and then a relaxation period, and we can measure that in milliseconds. And then that refractory period is the time when, we're not going to get into the difference between refractory period and absolute refractory period. We're just going to say during that time, another muscle contraction can't occur. We're going to build on that though. So the latent period, you have a muscle action potential that is going to go along the sarcolemma. We have to put a cell up here, because normally we were still, since it's been 4 days since we talked about this stuff, you have that muscle cell, and remember we have sarcoplasmic reticulum, I didn't mean it to look like a person, and then you have the T-tubules that come down in here. Just stay with me, I know it looks funny. You refresh, you're supposed to be picturing the actual picture in your mind, where that action potential sweeps along the sarcolemma, goes down through the T-tubules and back out, and that depolarizes those areas of the sarcolemma, which will depolarize the sarcoplasmic reticulum. That's what we're looking at. When that happens, you're going to force calcium to be dumped out into the muscle cell. And so at that point, calcium is going to be going out into the muscle cell. You're going to start to move troponin, tropomyosin out of the way like we know. You're going to start a contraction. And we're going to see that. So tension develops in that muscle. And then during the relaxation period, that's when the calcium is being pumped back into the sarcoplasmic reticulum. Right? I know I put all the things that are happening once calcium's in the cell and once calcium's back out of the cell. But I put them there just as a reminder of what's going on in the actual muscle cell that we talked about, down to that lowest sliding filament level. For some of y'all, this will make a whole lot more sense when we talk about how we actually build and conduct an action potential. We just haven't gotten there yet. We are, and before the exam, we will get there. And I'm going to show you how these compare. Right, but we're going to kind of continue on and talk about that second stimulus. What if another stimulus comes along? So when you have an action potential, stimulate a muscle cell, it's going to respond. If that action potential stimulates it, you will get this contraction. And then you would return to normal. Wave summation is a process to where if you stimulate muscle cells, a motor unit, you'll get a contraction. And if you stimulate it again before the relaxation period is over, actually, if you stimulate it again anytime after the refractory period, before it gets back to its latent period, you will get a stronger contraction. It'll contract more. And you could actually do that again and again until you reach max. So the second wave is added to the first. It's called wave summation. Another way to look at it is, in this case, calcium was released here. Calcium was released here. If we stimulate it again outside of the refractory period, we will release more calcium into the cytoplasm and cause more of a contraction. It goes all the way back to our original analogy where I was out here saying the second row. We're going to dump some calcium, they get closer. We dump more, they get closer, more. We can activate, put them together. The body itself, though, is going to use as least energy as possible to perform whatever action you're trying to do. It's going to do that through motor unit recruitment. It's going to take you start to lift something, and it's going to start with one group of motor units, and it will recruit more if it needs it. It's going to increase the amount of motor units used based upon the need. So that's going to come from the brain and spinal cord, right? So the more neurons that are activated, the more motor units will be stimulated. The more muscle tension that will be produced. And then you read the next one, we'll see that not all motor units are recruited simultaneously. All that means, I once hit somebody with a Dr. Pepper when I was explaining this one, so I stopped doing it. Because I had a plastic bottle of Dr. Pepper sitting here, and we can use a piece of paper. That's somebody's tissue. Like tape, sticky. Or not. Exactly. You notice the second time I picked it up with my left hand. I'm right-handed. I'm less likely to touch my face with my left hand. You didn't know that. But makes me just thinking about it. I should yawn now. Alright. So the point is, if I go to pick this up, I'm only going to use the amount of motor units I need to pick it up. If I recruited, just to be kind of facetious or too much kind of proper, if I recruited every motor unit at the same time, not only would I probably not make it there smoothly, but as I went to pick it up, it'd be and it hit dude about where you're at in the first row, the Dr. Pepper bottle. I don't know how I got the arc without hitting the roof, but it got on. It was only a little bit in it. Didn't go well. Actually, it was fine. Okay, so we're going to only recruit the number of motor units we need, and the body also, somehow it knows which ones are the weakest, right? So it's going to recruit the weaker motor units first. The larger, stronger motor units will be recruited as needed. If you want to think of it in more of just a picture type way, we're going to use a motor unit that only is in contact with 5 muscle cells. If that doesn't work, we can use a motor unit that's in touch with 10 muscle cells. So some motor units are large. That's all. The brain decides, which is very interesting. Like the brain. The brain decides, because how does it know that? It's constantly monitoring the tension in that muscle. Or in that musculoskeleton. Interesting stuff, if you like exercise physiology. Right. Cardiac muscle. When I get to cardiac muscle, I realize that we're getting towards the end of the description of muscles, and we've got a few things to finish out before we get to the neuron. Right? So if you look at cardiac muscle and compare it to skeletal muscle, right? Well, skeletal muscle is voluntary, cardiac muscle is involuntary. Cardiac muscle, amazingly, is going to contract 10 to 15 times longer than a skeletal muscle, and it's going to do that for your entire life. Now, you're not going to use your bicep 24 hours a day, but your heart muscle is going to be 24 hours a day. So that's very unique and kind of different with cardiac muscle. Right? So forget slides. If I'm going to use skeletal muscles to run, I'm going to use skeletal muscles. I'm going to build it an analogy in my head. I'm going to use my skeletal muscles, and my plan is to run 100 feet, continue on and run 1 lap, and to continue on and run 26.2 miles marathon, all in that order. Remember back to ATP use and what we're using? Well, let's compare that to here. Okay, if cardiac muscle doesn't do the sprint. It doesn't do the 1 lap. It's always doing the marathon. So what do you need, and what I was trying to build to is, what do I need once I get to the marathon? Aerobic respiration. So guess what the cardiac muscle is doing its entire life? Aerobic respiration. If I'm going to run through aerobic respiration constantly, what do I need a high demand for? Oxygen. How does the heart get oxygen? I know y'all just studied all this. That's why I'm not posing it as questions. I'm just reading through it. If I need more oxygen in the heart, it would probably be good if I had some main arterioles that go to it called the coronary arteries. Right? It also explains why if we cut off the blood supply to my bicep, or let's decrease it, not cut it off. We decrease the amount of blood supply going into some muscles. They don't notice it as fast, right? And over time, you're going to. If I do that to the heart, what's going to happen? You're done, right? You're going to be in the hospital. You're having a heart attack, right? Okay, so all of that to say, the heart's going to do aerobic respiration to produce ATP constantly. If you look at cardiac muscles, I don't even need to say all this, because I know you already know this now. You just had 2 tests on talking about. Muscle fibers exhibit autorhythmicity. It's a hard one for me to say from East Texas. They can contract at the same time. I like to say it that way better. Because they have the gap junctions, because they're connected, the ion flow goes through there. I don't care if you know the normal contraction rates at 75. It can vary based upon the individual or the study you're looking at, or what your watch tends to tell you it is. I don't agree with the average heart rate my watch tells me something. It's not really that smart of a watch. Maybe a new one. Autonomic and nervous system, though. Okay. So, how much your heart beats, how hard, how fast, is controlled by both the autonomic nervous system and the endocrine system. Well, that's fine because that controls everything. Right? All body homeostasis is controlled by the endocrine or the autonomic nervous system. And because of cardiac muscle doing aerobic respiration the whole time, that means that the mitochondria will be larger also. That's another difference you see in cardiac muscle. You not only have more mitochondria, they're a little larger. And the last one we have to compare to is smooth muscle. Smooth muscle is involuntary like cardiac muscle, but smooth muscle, I like to say things different ways. Skeletal muscle people separate smooth and cardiac, like, well, they have some similar characteristics because they're both involuntary. And you're like, okay, well, how can we separate these? And in my mind, I go, okay, well, cardiac's called cardiac because you find cardiac muscle in the heart. Smooth muscle is involuntary also, but it's generally found in like the viscera. But another difference, if we went all the way back to the chart, is that cardiac muscle under a microscope looks like the same setup in the sarcomeres as skeletal muscle. It's striated. it has thick and thin filaments, and sarcomeres that are connected by Z-disc. Smooth muscle, if you look at it, does not appear striated. Because, I kind of got ahead of myself there. I'm going to say this and I'll back up. Because the thick and thin filaments, right, we have those plus what's called intermediate filaments that attach to these things called dense bodies. And dense bodies are very similar to Z-disc. Right? So dense bodies are only found in smooth muscle. I'm going to back up just to say, because smooth muscle is associated with things like your bowels, like your heart, are constantly working. Everything you ate this morning right now we know is getting pushed through the system using peristalsis. But it's slow rhythmic contractions, like a snake swallowing a mouse, right? Slow, if you ever watch that, you can actually see it. Slow rhythmic contraction. I don't like snakes or mice. I don't want you to go home thinking that. Just a good example, right? And it's moving that through. Well, that's constantly going, but it uses anaerobic. It doesn't use aerobic respiration because it's much slower contractions. You don't really need to do that. He's laughing, so now I'm thinking, I don't really care whether you think I have snakes at home or not. I'm just getting worse than that. Hey, so I went back to catch that, but we were talking about what they call dense bodies. Right? I'm going to say it two ways. When you look at smooth muscle, the actual sarcomere, that's a sarcomere. That's a sarcomere. Right? Remember, and keep our second row in mind. So our sarcomeres in the second row, all the sarcomeres in the second row would be holding hands, right? The Z-disc. They're connected, their hands are the Z-disc. They're holding together. In a smooth muscle cell, they're not like that. They're apart. They're connected. It would be, in the analogy, if we took every other person out of the row or just extended the row out, you'd have a sarcomere. They're a good example. Is you'd have one sarcomere here, one sarcomere there, and then they'd have a rope between them. So now they're not physically connected at the hands by a Z-disc. In this case, each hand would be the dense body, and between the dense bodies would be a rope. The rope is an intermediate filament, and it looks kind of like a hairnet to me, right, when you stretch it down. So this is a dense body. This is a dense body. So those two sarcomeres, are they connected? Yes, but they're connected by intermediate filaments. So in that case, what would you call the functional equivalent of a Z-disc in skeletal muscle? The functional equivalent in smooth muscle is called a dense body, because dense bodies are what connect the sarcomeres, and it does that by using intermediate filaments. So that when it contracts, it looks like this. Contracting down. That's why I said it looks kind of like a hairnet. And that's as much as we're going to talk about smooth muscle right now. Yeah.
So the intermediate filaments, sir, are the same thing as like dystrophin for skeletal muscle?
In the way they function, yes. I mean, dystrophin's more specific for that tension reducing component on the end of sarcomere chains so that you don't tear the wall. But I know what you're saying. Functionally, when you look at them, yes. Alright, that's smooth muscle. You got one slide on smooth muscle, that's it, besides comparing it to the others. Exercise-induced muscle damage. Y'all are about to get a lot more exercise-induced muscle damage, I read. Forgot about that till just now. They're changing the PT test again?
There's removing one.
That's the only difference? Oh, okay. I thought so I read they just getting rid of it. Throw it there. I don't keep track of that anymore because I don't do it. So. They made it sound here and make it a lot tougher. No? It's good that I got y'all to keep me up to date. I don't know. I think, I'm gonna say. I think you should be able to be randomly PT tested at any time and you must pass. I know. That comes from the civilian who's not doing it anymore, but I would have told you that on active duty also. Some of y'all know what I mean, others get offended by that, but I'm like, that's. It comes from history. It comes from being a platoon sergeant in the 25th Infantry Division and having people who had, it was drag on me to keep up with people who couldn't pass it. And I hate it. I'm like, you should be able to pass it all the time. That's where that comes from. If you got offended by all that, I mean, be offended, I guess, but that's not something negative, so. Okay. Yeah, I'm going to regret that later. Put it on the critique. Some of y'all agree with me, though. If you don't agree with me, that's fine. DOMS. Delayed onset muscle soreness. 12 to 48 hours after you did the muscle. That's a term that's been used for a long time for what they don't fully understand. We know that if you go out and you work out, strenuously work out, and you start to use up the products of what muscles use to contract and things, you can actually start to fatigue those muscles, but you also can really push that workout and you can start to tear actual muscle fibers. They can be repaired, but they believe, you notice there's a key word in here that although, or a key sentence, although not all contributors to DOMS are known. So what they think is microscopic damage to the individual muscle cells themselves is what you're feeling when you have that soreness. You know, the guy or girl, whoever that goes and drops down at the weight bench and says, yeah, hasn't lifted in 3 years, but says, I'm gonna put up 300 pounds, and they do it once or twice, and they look big, and man, they're gonna be hurting, right? You know they're gonna be hurting the next day. That's dumb. Which reminds me, yeah, I stay on track. Anybody into like wrestling, not college wrestling, like the one where they put on the show?
You mean real wrestling?
College wrestling, real wrestling.
Professional wrestling.
But now I've identified you as the. I know, dude. I've never understood how to decide who wins. I'm pretty proud to have that belt, but isn't it just written that it's your turn to win? It must be you gotta, I don't get into it, so I was thinking it must be you gotta have the right look, you gotta have the right attitude, you know, you work the way up, like getting an Emmy. I don't know. For the wrestling people in the room, I'm sure they get hurt all the time, but they are physically doing that stuff. I don't watch it. What I was gonna say about that is if you're into wrestling, go out to Bernie YMCA and work out, because the Von Erichs now live in Bernie and they work out at the Y. 2 of the younger ones. They all do. Don't think that I'm at the Y working out. My wife works there occasionally, so she sees them all the time. So you go out there and get you an autograph. She says they're there every day. They have nothing else to do, I guess. And she had no idea who they were until I told her. I had muscle fatigue, because I was up there. I knew who Carrie Von Erich was. Iron Claw. Some of y'all don't know anything about wrestling there for the 80s. The Iron Claw. They couldn't get that off their head, no matter what they did. You grind on. Sorry, I'm having a little fun, because I was looking really tired there as I was going. That's why I kind of got off topic for a while. Back. Let's cover muscle fatigue and then we'll take our break next week. You could have just broke. Muscle fatigue is an inability of muscle to maintain the forceful contraction that you need to prolong that activity. Alright, so let's take the second bullet there and just say, okay, you wrote all this big, busy slide, because that's what actually happens. And rather than, once again, don't make you a list, you've already learned what causes a muscle contraction, right? We've talked about action potentials. We've talked about acetylcholine stimulating receptors on muscle cells, kind of thing. We've talked about that you need oxygen for muscle contractions or some form of energy. We didn't talk about that lactic acid builds up and stuff, but all of the products that it takes to cause a contraction in a muscle cell, if you deplete them, you will reach muscle fatigue. Like doing push-ups and people start to shake, right? That's not the whole process there, but you reach with it. Keep going until you reach muscle fatigue and you can't do it anymore. Some people reach that very quickly though. I mean, I'm just getting in trouble today with my statements. Because it rolls us into, okay, well what if you have what's called central fatigue? This is where I should have left the joking today. Because central fatigue is a true thing where you experience a tiredness or a feeling that I need to stop doing an activity because you start to feel tired, like you want to stop doing that activity. It's called central fatigue. And they think that this is a protective mechanism to keep you when the muscles are about to become damaged. But most of this in this room, as much as I joke about exercising, is most of us know that there are a lot of people, like you're grading somebody. I'm using these examples just for fun. Even yourself, you're doing push-ups, and have you ever been amazed when you feel like you've reached fatigue and you can't do it anymore? But you get somebody yelling at you, and all of a sudden, or you get pissed because somebody says something, or they don't count one because the guy who can't do a push-up save their life tells you didn't go well enough and takes one away, and you just start knocking them out, right? You do 20 more or something. I mean, it's happened to me once, at least, but no. Well, you just passed central fatigue. So the Army or military itself, they're big. That's what personal trainers are supposed to be doing, getting you to go past that central fatigue. You can do one more for the core. Get past central fatigue. I don't even know if y'all know what that means, but I got nowhere else to go. Take a break. I finished that in a bad way that last hour. I was going good till then.
If y'all could please check the chat. There's some people listed on that 50. I don't have time to scrub it. Some of you are on there. You have to go 11:30 to room 0516, and I would not pee before you go down. So if you're on that list, you got to go down to 0516 would be away. I saw at least 6-ish of our names. And then the first sergeant meeting over lunch is now at 12:30. I'm waiting for sure confirmation, but it's got pushed back. So I'll keep updating in the chat.
Y'all get back right at 10 minutes. I'll get y'all out of here. Did I go too far on tracking of
because he sold the house, went to a basement, which was the size of the basement, and a bathroom, kitchen.
These are
The trailer.
But he was the final. I've done this many times. You're welcome to try it.
I love how, like you said, I paid attention to the garage door over top of the window handle. Okay, you don't close the windows. You ain't open. I mean, I really like this. There's 5 of them. Dude, you can eat all you want, and then at night you can just go around it. Like, dude, horrible. Yeah, that's awesome. That's funny. Life would be so long. Life would be so long. Yeah, exactly. Really, life would be so long. If you wanted to, for us to make it. What do you mean? It's fair. So tight, though. That's been a all of us, all the time. Send me a coach. You know, it's like whatever. That's all down. I think that's what I got. One of them was the pressure was the lowest, and then the other one got wrong.
I think the resistance from those much from the hard part is.
Oh, look, different Jay over there. Yeah, we're gonna take the master. I don't remember. I mean, I'll be up to one because I got wrong. Yeah. That's right. I get more annoyed about I want to put it together. She wants to put the one of Jason. She's afraid to come. This is fun.
The other side.
I don't know what basketball are. It's got a sign on it. I already have sent it.
I was wondering at first, man, I used to do 100 They have to make sure
That'd be great.
I remember there was like a this.
I don't know which one it was. It might have been down where it said like it was this Rob's which one of these no, not that. That was the name which has the least version. Yes, no, but I think the one they're talking about it said like if consistent drop, you know, like take everything else is the thing is, yeah, I did that too. There was too much time in this way. Back to fix that. One reason I was like, I calculated
I calculated right, and then I calculated wrong. The first one was -2, and I was like,
I picked that one.
I put -2, and then I went back.
With the highest and lowest for this test was like 8 times. Yeah, lowest was 2. What?
Alright, here we go. Alright, what about muscle tone? Tone. Everybody's heard of muscle tone. Muscle tone is small amount of contraction in all your muscles that's happening right now. Maintain posture, to maintain you from crapping on yourself, sphincter tones, all of this. What's interesting about muscle tone, people often go, okay, there's a little bit of contraction in every muscle. You were to sever the innervation to that muscle, it would become flaccid. All the muscle would relax. Okay? Muscle tone itself is set from looking at areas of the brain and spinal cord. And there's a skeletal muscle tone and there's autonomic muscle tone. Those are two different things. But muscle tone is set by neurons in the brain and spinal cord. And what it's doing actually is you look at the different motor units that one bicep has and it's creating the tone. It's not just using one motor unit, it actually cycles that, and you'll use a small motor unit, different ones at different times, but you'll constantly have one of those in a small amount of contraction. muscle tone. And it's very necessary, like I said, to keep things moving through the bowels, to keep your posture upright, your head up. Well, what if we talk about all that and we say, well, what if we have a problem with the actual muscles? Anything that affects the motor neuron, in this case somatic, every time you think somatic, think skeletal muscle, think voluntary. Somatic motor neuron, the actual neuromuscular junction, or the muscle fiber itself. In other words, we're going all the way back to one somatic motor unit and saying anything that affects the neuron that's here, the synaptic cleft between that, the exchange of information that happens there, or the individual muscle cells, is a neuromuscular disease. Right? We have many of them. One of the most common, I just put one, I didn't go a lot into these, I'll put you some more definitions after this, I'm sure. Myasthenia gravis. Well, myasthenia gravis is a very interesting autoimmune disease. What it does is it creates antibodies that block acetylcholine receptors. What's the one, if I was to ask you a neurotransmitter, and think of, see, I've already learned what I said, acetylcholine. But for the class, all skeletal muscles use acetylcholine as a neurotransmitter. So if I block the receptors for acetylcholine, I essentially cannot stimulate muscles. That's one of the things that does that is myasthenia gravis, where the antibodies that get produced have the ability to bind to those acetylcholine receptors, so they're not available for acetylcholine to bind to them. You do that long enough, the muscle will atrophy and you'll lose function of it. All right, the definitions are yours. Just a bunch of definitions on different types of muscle contractions, small and large, just so you kind of get it. I got kind of carried away. Good things to know though. They will come back up in many classes. So that's the end of muscle, and we are going to go a little bit into neuro today. How far that is, we'll see. I'm not going to get too much into it. We're going to look at the basic setup. So nervous tissue is where we're going to get action potentials. Say that, where we're going to generate action potentials. Nervous tissue is part of maintaining homeostasis, we said, because of the endocrine system and the nervous tissue. Okay. Nervous system, the reason it's repeated here again, is now we're going to say, well, if it's the nervous system helping to control homeostasis, that's going to happen pretty rapidly. If it's the endocrine system helping to control it, which they're both doing this all the time, but the endocrine system is going to be slower because it has to move the hormones through the blood to do that, or cell to cell at least. When we look at our breakdown of where things are located, remember that the central nervous system is the brain and spinal cord, and everything else is considered the peripheral nervous system. So in that peripheral nervous system, we're going to have the 12 infamous cranial nerves. We're going to have spinal nerves that have 31 pairs, and then we're going to have just peripheral nerves. In other words, all the rest. And the nervous system itself is broken down into sensory, integrative, and motor. All right, so you're going to have sensory receptors. They're out there, there's a lot of different kinds. They're detecting the difference between your internal and external environments. Right? So the sensory cells can carry that sensory information from the receptors to the brain and spinal cord. Remember our feedback loop slides? So the sensory receptors are carrying the information back to the central nervous system. Inside of the central nervous system, you have neurons that can analyze and make decisions about information. And then it's going to send a response to an effector by sending information down a motor neuron. So sensory neurons are always ascending, carrying things to the central nervous system, and motor neurons are always going to be descending. Right? Sensory neurons are also called afferent neurons. So afferent neurons are neurons that go back to the CNS. Efferent neurons are going away from the CNS. Those are two words I underline them because you need to be familiar with them, even though it's easier to say. Sensory neurons, ascend. Right? Well, ascending is an afferent neuron cell. The other way we broke this out is to say, well, there's the somatic nervous system, there's the autonomic nervous system, and then there's the enteric. So somatic doesn't just include skeletal muscles. So we haven't talked about all the sensations yet. So sensations out there by those muscles, so the limbs, the head, your special senses. And there's a big difference between saying, like touch is a general sense. Smell is a special sense. Matter of fact, we spend a whole testing block just talking about special senses. And they're different, but they're under somatic. Right? And then motor control of skeletal muscles. So what I was saying last hour is true. When you think somatic, think of skeletal muscles voluntary. But there is sensory coming from those areas around those muscles. That's your somatic sensations, along with special senses. So we're going to get really deeper into that. The autonomic nervous system has sensory receptors that can tell how much stretch is on your bowels, how fast things are moving, what's the chemical composition inside of that area. And then it's going to send motor neurons back out to those involuntary muscles, glands, cardiac muscle, whatever it might be that it needs to control. And then the enteric nervous system is interesting because the enteric nervous system is controlling the GI tract. And at one point, before they figured all this out, they gave the enteric nervous system a different name. I say they because I don't know who it was, but so there was a thought process long before any of us that the bowels functioned independent of the central nervous system, that they weren't connected. Now we know that's not true now, because if you look at somebody with a spinal cord, if you have to get information to the central nervous system, and let's say I cut my spinal cord at T4, thoracic region 4, did I sever the pathways for that information to travel to the bowels? Whether you're looking at sensory motor, yeah, I did. I completely cut it. Do the bowels still work? Yes, they do. Their enteric nervous system will function on its own without its connection to the central nervous system. So then why do we care? Well, if you were to go and look at like a spinal cord patient in that situation, and I say their bowels still function, do they function well? No. They're going to be doing bowel programs. They do function to an extent, but they're going to need help. They're not optimal at all. Right? Well, we can also throw in there, well, the bowels got that name. They didn't do that with the heart. Because guess what? If I sever the innervation of the heart, it has an intrinsic way to keep beating too. I don't know if y'all find that interesting. I do. I don't want to have that happen, but. So the enteric nervous system, to me, I like to consolidate things. Like when I clean and things, let's consolidate, label, get it organized, and it makes more sense to me. I think it's time. I'm making a pitch here. I think it's time to just put the enteric nervous system fully in the autonomic nervous system category. Just keep that in mind. One of y'all do that later, because I'm checking out here in a couple of years. I'm not going to get involved. I don't know when that year is, but sometime between now and death. So, I get that question a lot. Because I've been around a lot. They say, "Oh, when are you thinking about leaving?" They say retiring. I don't. If you're working, and every day you come in, retirement's on your mind, and you're not like a week out from it, stop. That just ruins your life. I hate that question. When are you going to retire? I don't know. I'm very serious. I told Bond, and he came in today. How you doing? I said, well, I kept going up the steps. Every day I get to the steps, and he looked at me funny. I said, you don't ever get to the steps and go, all right, I'll start these steps and going. If not, I can turn around. I can say, screw it right now and go back to the car. Then he walked out, and I figured out why he didn't get it as much as I said. He kind of chuckled, but I don't think he got it. It's because he's still in the military. But if he turns around and goes back, somebody can do something about it. If I turn around and go back, not a dang thing they can do about it. You got to think about these things to keep yourself happy. You have control of something. You know. I'm going to keep coming though, as long as y'all keep laughing at my dumb jokes. Okay, the nervous system. Another way to, it's a different type of slide looking at it. All I'm trying to show you here is we need to know where things fall. We understand already. I know that, oh yeah, somatic, autonomic, we've seen that a million times. Okay, we got it. What we're going to focus in on, and we repeat, to say the somatic nervous system has somatic and special sensory receptors, and then somatic motor neurons are voluntary and they go to skeletal muscle. The central nervous system is connected to all of these. Autonomic also sends sensory information to the central nervous system and receives autonomic motor neurons to your organs, you know, vessels, organs. And that will talk to also the enteric nervous system. Okay? And the enteric nervous system actually does send sensory to the central nervous system to tell it what's going on in the bowels. Now, one of the other areas that we're going to spend a lot of time on is sympathetic and parasympathetic systems. So the whole sympathetic, parasympathetic, fight or flight system resides, and what I'm trying to show you here, all I'm trying to show you, is that part resides in the autonomic motor neurons. Okay? Because we're going to come back and piece all this together and look at, well, what do you mean by that sensory receptor that's going there? We're going to get down to that. This is just kind of the overview that you have to understand before we start doing that. I didn't leave these as definitions because they're too important, meaning you got to know the differences in these to understand talking about neurons. Right. If you're looking at the peripheral nervous system, everything outside of the brain and spinal cord is peripheral nervous system. If you have a collection or an association, somebody last class, now been out of shape just meaning they asked me, and I call it been out of shape. And they're like, I was talking about something and I said, well, you have a collection of neuronal axons in the peripheral nervous system is called a nerve. And I said something else and they're like, we had a disagreement about something with that. Oh, they're like, you said a collection, but now you're saying an association. Those always strike me, I'm saying it, because that strikes me as really funny when people say things like that, because I'm like, okay, you know, you just nod. Yeah, we're talking about the same thing. And they're like, well, no, a collection is not, this is what got me with this. A collection is not an association. I'm like, it's not? I'm like, no, y'all look at me like you don't agree with me. Like, I might ask you that. How do I know, why were we talking about this? Because I asked it on the test. He was trying to say a collection and association are two different things. And I wasn't buying it, but anyway, that just felt flat. That's why I said, okay, let's see why this is important though. How about that? So, an association or a collection of neuronal axons in the, we haven't studied neurons yet, but the axon is part of the neuron. If you have a collection of axons in the peripheral nervous system, it's called a nerve. If you have a collection or a group of neuronal cell bodies, the cell body of that neuron, that's called a ganglia in the peripheral nervous system. And in the peripheral nervous system, we also see a plexus. A plexus is like a spaghetti mixed up with nerves or veins. Right? But if we switch to the central nervous system, and I didn't do it, but it may help to write real big on the top of the slide, PNS, because all of these are peripheral nervous system, because you're going to have a difference in terminology naming. So now we have an association of neuronal axons in the central nervous system. You will not call it a nerve, you will call it a tract. If you have a collection or association of neuronal cell bodies in the central nervous system, now it's called a nucleus. So these are compare and contrast. Peripheral nervous system, you have a nerve and a ganglion. Central nervous system, same looking things, but now we call it a tract and a nucleus. That will always be the case. Histology of nervous tissue. What does it look like? An actual neuron has cells that can actually produce electrical excitability or action potentials. They can prepare and transmit action potentials. In other words, how do we stimulate muscles and glands? Well, we use nerves. If you're looking at the nervous system and you have a cell that is associated with the nervous system, but it's not a neuron, it's called a neuroglia. So there are many types of neuroglia that support and nourish and help these actual neurons. There are a lot more neuroglia than neurons. Neurons are not replaced. You can study all you want, you're not making new neurons. Right? So you go out and huff paint, and you kill your neurons, you're going to develop just more neuroglia, which aren't going to help you. You're going to build more support cells for the few neurons you have left if you huff paint. Every time I go to Walmart, Home Depot, anywhere, I have to show an ID. Every time I have to show my ID. Every time, I don't know what, I guess I only buy stuff that requires an ID. I don't get that. I know. Paint.
Whole lot of work.
Spray paint. People huff it. Huff that. That's why I use it. They're killing it. You didn't know that? People take acetone, all sorts.
Oh, yeah.
They,
I know they have some paint, but it's not back. I don't really do that.
I guess I'm gonna start telling the huffers to move to Colorado.
They're already on there.
No, go to Walmart. In Bernie at Walmart, or Bernie Home Depot, sorry. You can go get your paint, go to the cash register, and just start splashing. Kind of like, and Walmart's the same way. You go to the Walmart, Home Depot down there by Redmond Road, it's Fort Knox. You can't even buy a car charger without asking somebody to unlock it. And they lock all their paint up and they get you the register too, probably. I don't know. I'm always buying car chemicals, so every time I go in there, they're flagging me. I'm like, dude, you see me every day. Can we skip the ID? Yeah. Good way to get me digressing there. No. Typical structure of a neuron. Neurons are a cell, but remember, muscle cells didn't look like normal cells or generalized cells. Neurons definitely don't. Neurons are very unique shape, and that they're going to have a cell body. Then they're going to have an axon, generally just one axon. Sometimes you can have additional. That's the axon. And then they're going to have dendrites. These are nerve fibers that come off of the cell body. We're going to change some names up for us. Because remember, it can't be easy. We got to make different names. We got to say plasmolema. Then we got to study muscle and say sarcolemma. And now we got to change it and say axolema. Truthfully, it makes sense, so it lets you know what you're talking about. We got general cell, muscle, and neuron. And of course, we have axoplasm also instead of cytoplasm. Even though when we look at neurons, they look different, they still have all the normal cell components. A little bit of difference though, you have Golgi bodies, lysosomes, mitochondria. But what's unique about nerves, neurons, not nerves, that's not the correct terminology, right? A neuron cell body, when you look at it, the rough ER isn't right there by the nucleus. It's broken into clusters all throughout the neuron, and it's called Niesel bodies. So Niesel bodies are prominent clusters of rough ER, and they're gray. Right? So, if I can find some. Niesel bodies are going to be found all through here. Because of their gray appearance, any place in like the central nervous system where you see a large collection of neuron cell bodies, that will appear gray instead of white. White, we're going to see as tracts with myelin and stuff. That's an interesting part of neurons. Then you have the typical structure, the dendrites, which means these, the little trees that come off of them. This is the receiving portion of the neuron at the dendrites. There are different connections we have to go over about what connects dendrites. The axon part is what's going to send the signal that comes out of this cell, and it's going to propagate, you'll see, down the axon. So just for reference, I put they can be very long, like the vagus nerve, it's like 3 feet long in humans, that's very long for a nerve. What would, like if we're innervating a skeletal muscle with a neuron, the muscle would be here. You'd have your neuromuscular junction, and at the end, we have those synaptic endpoints. Just referencing back to what we talked about. So generally, you only have one axon. That's what we're going to study most of them that way. And typically, that axon is going to arise from an elevation in the cell body called the axon hilli. So that's this region here, where the axon comes off. And part of that is called the initial segment. It doesn't matter exactly where it's at. I'm not really going to ask you to measure or to label the initial segment versus the axon hilli. But there's an initial segment. That's important because in most neurons, not all, the initial segment is where the action potential starts before it propagates down the axon. Underline generally, because this area at the initial segment is actually called the trigger zone. In other words, where an action potential starts in a neuron is called its trigger zone. But the trigger zone is not always here at the initial segment. It is in most neurons, but I'm going to show you a difference. That's why I said double underline generally, or something to key you in that. He said this for a reason. I'm going to show you the reason here in just a second. Right before we finish out today. I'm going to come back to it. Axon, we said, contains normal components. So we have the mitochondria that are there. Whoa. Killing me. Microtubules that are there, neurofibrils, which are, we'll talk about that in a second. They're found within this actual, we said it doesn't contain rough ER. So when we look at, sorry, we're talking specifically about the axon. When we look at an axon, it doesn't have rough ER. What do we make at the rough ER? Proteins. Or we can make proteins, neurotransmitters that we might need. Like we can make acetylcholine. The axon doesn't have the ability to do that because it doesn't have a rough ER inside of the axon portion. That's what the slide is saying. And so protein synthesis and things have to occur up in the cell body. And what we'll see is we're going to send that information from the cell body down. Occasionally, not always, not even very commonly, you can have axon collaterals, where another axon branches off of an axon. Most of the time, we're not going to talk about it that way, but it can happen. The neurofibrils that are in the axon are really intermediate filaments. You notice we use intermediate filaments for a lot of things, right? To make myofilaments, to strengthen things. There are intermediate filaments, bless you. Intermediate filaments that provide shape and support to the axon, give it its structure. And then there's microtubules, which are another component of cytoskeleton normally. Microtubules that participate in moving things between the cell body and down the axon, even all the way down to the ends of it. So the microtubules are going to help do that. They're like a train track going back and forth. That's called axonal transport. Right? When you get down to the end of the axons, we call them synaptic impulses. The synaptic impulses are the very large part, but you actually have branchings in their axon terminals. So you're going to make new proteins, vesicles, whatever you need and use in that cell. You're going to make that in the cell body. You're going to transport it down to the axon terminals, which may be, like we said, 3 feet from each other. So you need a way to move those. So it's going along the inside of that axon. And the way we're going to do this is called slow axonal transport and fast axonal transport. Slow axonal transport only goes one way. It goes from the cell body to the ends of that axon, the axon terminal. Right? Generally, slow axonal transport is used to help axons regrow or grow. Okay? In order to, I often picture it this way. Cytoplasm is needed as an axon starts to grow or regrow. And I picture cytoplasm in a neuron being like honey. And if you were to pour it, it's going to roll slower. So it's not going to go very fast. Okay? But it needs to reach that axon. So it's a slow process. Fast axonal transport, though, can transport things either way. Right? It can go down the axon to the terminals, or it can go from the terminals to the cell body. And it uses the microtubules that are in that axon. Right? So it's using the train tracks to go back and forth across. It makes it very faster. You're going to see that we make things like synaptic vesicles, synaptic end balls that can use that way to travel. Okay, and really kind of the last thing we're going to talk about today, we're going to finish a little bit early here, apparently, but is where I was going to. We're going to look at the structural classification of neurons. So in different areas of the body and different components of the body, we find that the neurons look different. Almost all of the neurons in the body, not all, are multipolar. Right. I have to say this: multipolar, bipolar, pseudo-unipolar. Sound like it might hurt. The way your neurons are classified, you never thought about this probably, but you have a cell body. You go in and count, and if you see multiple dendrites coming off of the cell body, that's called multipolar. If you have one or two coming off, then change the name. So a bipolar neuron, bi meaning two, has one single dendrite. That's not working. Now that's it. You have one single dendrite, and then you have one axon coming off it. So because there's not a bunch of dendrites, they don't call it multipolar, they call it bipolar. It's a bipolar neuron. And then when you look at pseudo-unipolar neurons, they actually only have one real connection to the actual cell body. They look very different. And I'm going to explain to you the difference, and then we'll be, once we finish these, I think we're done with it. So a multipolar neuron, I was just trying to show you the way they're named. They have many different dendrites. Mostly all of the neurons in the body are multipolar. The dendrites, if you picture, are receiving information. And so we already know that neurons talk to neurons and other neurons. And so really you would have normally you have a bunch of neurons that are talking to this one neuron. And it's talking to other neurons, right? It's tracks, they're connected. That's a multipolar neuron. In a multipolar neuron, the trigger zone is at the axon hillock, or the initial segment. But even though we haven't studied action potentials in detail, that's where the action potential starts, and it's going to travel down the axon. So in a multipolar neuron, the trigger zone is found here. In a bipolar neuron, they have a trigger zone that is also at the axon hillock. Remember, we said there's only one dendrite. Now you look out here and go, well, it looks like a lot of dendrites. That's not the definition of why we name it. The definition says how many connect directly to the cell body. And in this case, just one does. So that's definitely a bipolar neuron. Right? When we look at special senses, it's the only place we find bipolar neurons. So if you look at things like the retina of the eye, the inner ear, the olfactory area of the brain, when we study those, we're going to see that some of them use bipolar neurons. So bipolar neurons are specific to be associated with special senses. The reason I'm saying it that way is because when you look at a unipolar or pseudo-unipolar neuron, right, it actually has a trigger zone down here. This is not going to make sense to a lot of people, but the trigger zone's here. I'm going to explain how it's set up, not how it works that way. Right, so pseudo-unipolar neurons are used for general senses. Bipolar neurons can be used for special senses, but pseudo-unipolar are general senses. Okay, well the trigger zone in the pseudo-unipolar neuron is not at the axon hilli. It is found out by the dendrites, which are out here. So what happens in a pseudo-unipolar neuron is it has one main line coming into the cell body, and then the motor fibers leave out of the same area. So it looks unipolar versus bipolar, right? Only one connection at all. The part I want to explain, and then we're going to be done for the day, so we're going to stop here at this side, is because the triggers, I'm going to list one of those things where I talk about it just has to be that way. It has to be a fact. It has to be what they found. Okay. A general sense is like touch. I'll use a skeleton. So, when you look at special senses, great. That's in, you use bipolar neurons, that's up in the head. So almost your special senses are there. But a general sense like touch is like out here at your fingertip. And so if you touch the fingertip, right, I have to send the signal from there to my central nervous system. And this is the part that we don't know yet. All of these pseudo-unipolar neurons that are used for general senses, their cell body is located here. We're going to learn that that area is called the dorsal root ganglion. So all general sensory neurons, all of the sensory that you have coming in from, let's say, this whole hand, has very, very long connections all the way back to the cell body that's here. So if in multipolar neurons the trigger zone or the start of an action potential is at the cell body, that wouldn't work in this situation, right? So the trigger zone's out here by the dendrites. And then it has a very long process that extends back to the cell body. So the trigger zone's out there. Because if I ask you that, you're going to go, well, if you're later after this, because you're not going to remember that, you're going to go, well, the trigger zone's at the cell body, axon hillock, initial segment A. Well, it is. Most neurons are multipolar, and that's mostly what you see. But in general sensory neurons, if they use a pseudo-unipolar neuron, it will be found out by the dendrites. It's in a different location. Don't ask me why. No, go ahead.
That's because the dendrites in this case are actually receiving the signal.
Yes. That's because the dendrites of a neuron are the receiving side. The axon is the sending side. I mean, I'm joking with you in a second, but I appreciate you saying that, because some people may not realize that the dendrites are always the receiving side. So the touch receptors out here, when I say I have the feeling of touch, what I'm really saying is that there are free nerve endings and things down here on my finger, free dendrites, that are the sensory receptors. We're going to get into that in neuro, but, right? The free dendrites that pick up touch are the sensory receptors, or what it's called the dendrites. And they will send that signal back to the cell body, and then it will go on into the central nervous system. That makes sense? If it doesn't, because always, you need it for other classes. If it doesn't, go back to this slide and say, the trigger zone is here, the trigger zone is here. Both of those are by the cell body, and a pseudo-unipolar, the trigger zone is here instead. Just the difference. The only other thing I needed you to know is that most neurons that are used are multipolar, bipolar, special senses. Pseudo-unipolar, general senses. Right? Are we going to get a little more detailed into that? Yes, but that's the start.
Sir, could you just maybe briefly re-explain what the trigger zone does again? Is it just, it says it starts the action potential, or?
So, no, you're good. I'm going to go. Okay, maybe I'm not. I saved it for the next one. Okay. I needed something blanker. Let's do this. Here's a neuron. We're going to use this multipolar neuron. Neurons will have other neurons. Right? Let's put some dendrites out here. Neurons talk to neurons, muscle cells, and glands. Right? So I'm going to just simplify. You have another neuron, another neuron. So they're all talking to that one neuron. So if this neuron sends an, we are going to discuss this tomorrow, or on the next test, one of the two. I don't remember. But it helps too. Each one of these is sending an action potential down to this neuron. Okay. The part we haven't discussed is when that neuron cell body receives all of that information, it has to take that information and sum it up and make a decision whether or not I'm going to fire an action potential. If it does do an action potential, or if, let's put it in layman's terms, if this neuron decides I am going to send an electrical potential down my axon, when it makes that decision, the trigger zone is where that action potential starts. Now, I could have just said it that way. The trigger zone is where all action potentials start. The problem comes from, which we haven't discussed, what do you mean by action potential? And actually you have, because you talked about it in cardio. Did y'all talk about threshold? Okay. When, in order to send a signal down a neuron, you must reach threshold at the beginning. And that's going to occur generally, or always at the trigger zone. And normally the trigger zone is by the cell body, except in pseudo-uniform. So the trigger zone is where summation occurs. All of the info that neuron receives. Because what we don't know is that some of the stimulus coming from those neurons that I drew, some of that's going to be excitatory, some of that's going to be inhibitory. Is that helping? I'm getting way ahead of where we're going. But so once the neuron makes a decision, though, the first segment of that neuron will be depolarized, as in cardio, at the trigger zone.
And sorry, just to follow on, what's the point of having it further away in a pseudo non-polar? And why would you want that to happen further away, down closer to the dendrites, opposed to having it close to the cell body?
I love
am
it.
I getting into tomorrow? If not,
No, we're not getting into tomorrow because we're never going to know that.
Okay. We don't know.
No, no. They say I thought he was going to ask, don't ask me why. Is that's the way they're set up. I don't know that there is a why. I like to go back and think of, like, if we were the ones studying these neurons and they mapped it out, what they figured out is, think of it like they studied and said, hey, look, we're going to call this multipolar. And they figured out how they did it, I don't know, but there is a why for that. But they figured out the action, the signal that went down this started here. And they saw these and went, well, these look different. Oh, but the signal still starts here. And then they came out and studied other ones and said, oh, look. And now, let's picture you're here and you're like, I figured it out. You do it, I'm being 1883 or something. I don't know how long ago it was. You're like, hey, we don't just have multipolar with all these dendrites. I found a neuron that has 2 connections and only 1 dendrite. So I'm going to call that a bipolar neuron. But hey, guys, don't worry. It's no different. The action potential still starts here. And then some other yahoo comes along and goes, well, that's great, but now I was studying and I actually found there's a neuron that looks different than the other 2. So we had to come up with a new name. What should we call that? And he's like, well, it only has 1 connection to the cell body, and y'all decided to how many dendrites connect to the cell body to name them. So we'll have to call this unipolar or pseudo. I'm not going to get into what it's really. Anyway, pseudo unipolar. There's a whole talk there too. And he says, yeah, but my neuron's different. Because y'all said it always starts at the axon hillock, the trigger zone's always there. But what I've found is that's not true. Because in order for this, I noticed that the axonal starts here. It's still doing summation. There's no cell body there. This cell body is always by the dorsal ganglion, in the dorsal ganglion. I said all that to say, to walk you through, like, they were mapping it out. They're like, that's just what they found. I think the why comes from, maybe I'm wrong, if somebody knows more. I don't know how you would ever know. We can reason out why it is, but why it was built that way depends on your religious beliefs. I don't know. You know what I mean? Or non-religious beliefs. I don't know. It just is. Anybody disagree with me on that? No more? Because it's just the way that's what we knocked out and saw.
Well, I would say the trigger zone needs to be close to the dendrite so that if I stick my hand on something hot, or a tiger bites it, or a spider bites it, I know to pull it away as quick as possible rather than just leaving it there.
He's making another point that we will get to. That part is how fast that's going to occur. And if that trigger zone is not out there, not only could you still send the signal? Yes, because you have this very long, just dendrite thing. And that would be a reflex reaction. But since the trigger zone's out there, right, it's going to be much faster. Still didn't explain your question. I mean,
that makes sense, and that's what I kind of assumed. I'm just trying to differentiate why it's.
It's kind of a funny way to finish. Just mind drop again. Y'all go eat lunch, man. I'll see you tomorrow.